Good afternoon and a very warm welcome to all the respected professors and the enthusiastic audience. It is a privilege to introduce a scientist whose work has profoundly reshaped our understanding of who we are, where we come from, and how our genetic past continues to shape our health today. We are deeply honored to have with us Dr. K. Thangaraj, one of India's most distinguished human geneticists and currently a CSIR Bhatnagar Fellow at the Centre of Cellular and Molecular Biology, Hyderabad. In recent years, national initiatives like the Kashi-Tamil Sangamam have beautifully celebrated the cultural, linguistic, and spiritual bonds that connect diverse regions of India. What culture has long affirmed, modern genomics now powerfully echoes. For over 3 decades, Dr. Thangaraj has been the forefront of population genomics, medical genetics, and ancient DNA research. His scholarship has illuminated some of the most fundamental questions about the peopling of the Indian subcontinent. Dr. Thangaraj's research has shown that despite an immense linguistic, social, and geographical diversity, Indians are deeply connected at the genetic level. Dr. Thangaraj has published more than 300 research papers in many of the world's prestigious journals like Nature, Science, Cell, Lancet, etc. He has served on editorial boards of major international journals and has been elected fellow of the Indian National Science Academy, the Indian Academy of Sciences, and the National Academy of Sciences. His accolades are too numerous to list, but they include the J.C. Bose Fellowship, Sun Pharma Research Award, Sir C.V. Raman Memorial Lecture Award, Sir U.N. Brahmachari Award, multiple Lifetime Achievement Honors, and several distinguished oration awards. Recently, he was also awarded with the prestigious Vigyan Shri Award. He has also led major national and global initiatives, including the Genome India project, which catalogs the genetic diversity of India's many endogamous communities. His pioneering studies on the Andamanese tribes provides the first genetic evidence supporting the early migration of modern humans out of Africa. His landmark work in 2009 unveiled the dual ancestral components of the contemporary Indians, the ancestral North Indians or the ANI and the ancestral South Indians or the ASI, reshaping the long-standing narratives about our population history. Through his analysis on endogamy, founder effects, and population-specific disease burden, he has shown how history, culture, and genetics intertwine in the Indian subcontinent. And through ancient DNA studies from the Indus Valley civilization to the mysterious skeletons of Rukun Lake, he has helped decode chapters of our past that were previously inaccessible. Beyond his scientific contributions, Dr. Thangaraj is a visionary institution builder, former director of the Center for DNA Fingerprinting and Diagnostics, former director of INSTEM, and founder president of the Society of Mitochondrial Research and Medicine. Today, we have the rare opportunity to hear from a scientist whose work has not only advanced the academic knowledge, but has also shaped public discourse on ancestry, health, disease, and ancestry. So before we begin with his talk, I would like to request our head of the department, Professor Thangaraj sir, Professor Lakhotia, and Professor Raman to please come on stage and felicitate our revered speaker.
You don't need to sit.
So, on behalf of all of us gathered here, it is my great pleasure to invite Dr. K. Thangaraj to the stage to deliver his lecture on a special talk series on archaeozoology titled The Genomic History of the Indian Subcontinent. Over to you, sir.
Thank you very much for very extended introduction. Also, you summarized my talk. Yeah. Thank you, Narishwar, for inviting me to talk to the students who have elected archaeozoology. I think it's a very important topic, not only to study as a subject, but also understand our history and our future, health, disease, and so on. It also gave opportunity to meet my teachers, Professor Lakotia, Professor Rajram, and dear friends, and many others. So what I'm going to talk to you maybe 1.5 hours, maybe it is more than 1.5 hours, is that how the Indian populations have arrived here and how our culture, particularly the endogamous marriage, has led to a lot of population diseases. Very briefly, and towards the end, I'll also talk about ancient. Before going to the subject, I also wanted to highlight that why India is very specific, why we need to study Indian populations. To begin with, I wanted to just highlight a quote of Mark Twain, is an American writer. So what he said about India? He said, India is a cradle of the human race, the birthplace of human speech, the mother of history, the grandmother of legend, and the great-grandmother of tradition. Our most valuable and most instructive materials in the history of man are treasured up in India only, right? So we'll understand that who lived during late 18th century and early 19th century, as how he is predicted without knowing much of our genetic work, because he could comment about it. The second one, why we need to study our history, I also want to highlight the work of Vigyan Shri, one of the well-known evolutionary biologists. He said that nothing in biology makes sense except the rate of evolution. Again, you will understand as my talk goes that how important this statement is, and why studying evolutionary biology is very, very important. But again, I wanted to quote another paper of Vigyan Shri. When you talk about evolution, genetics play a role. So we cannot distance genetics and evolution. So whatever the genetic change actually brought a new species or new phenotypic features and so on. Even if you look at any disease, sickle-based mutation can change drastic changes. Therefore, genetics is very important. We cannot distance genetics with evolution. Variations play a major role in evolution. Actually, this paper, I don't know whether you read or not, what he has mentioned is that type is the raw material of evolution. What he says, in the first paragraph you see, for nearly 80 years, evolution has been one of the basic problems of biology. During this time, however, the problem itself has undergone a most significant change. And I mentioned about Darwin's work. Darwin and his immediate successors had to prove first and foremost that evolution has actually taken place in the history of Earth. He said 80 years. That means he's referring this book of Darwin, who published in 1858, The Origin of Species. where he has highlighted about origin of most of the organisms, right from Archie to Whale. But what was missing in that book was how human must have originated. Around the same time, another evolutionary biologist, Thomas Henry Huxley, in fact, he is the first one to propose that anatomically, the modern human is similar to apes and other non-human primates. This is the first book which was devoted to human evolution. We brought the evidence that the evolution of human and apes have come from a common ancestor. You can see it was published the year 1863. But 8 years later, again, Charles Darwin published another book called The Descent of Man, where he has actually declared that chimpanzee and gorilla are our closest living relatives, again based on the anatomic similarity. That time there was not much of genetics was known. But now, after human genome sequence and many other organisms sequence, it's very clear that we, the modern human and chimpanzee, have originated from a common ancestor called hominids. Right? I don't know how many of you believe on evolution, but one of our ministers goes to higher education, and he, in fact, you look at like his statement came in Nature, but you can see if I zoom in further, say that the one which is underlined, I know that you can read it was down. Darwin's theory of evolution of human is scientifically wrong. And he said nobody, including our ancestor, in written or orally, have said they saw an ape turning into a man. Okay? The evolution is not like a kind of magic that chimpanzee or ape just by a second or few minutes turning into a human. It is a long process, as all of you are aware. It took about 7 million years to evolve the way we look like today as a modern human. In this process, there are several species have evolved, and most of them have vanished. None of them are surviving. So in this process, there are several phenotypic features, several ability we all gain. So one of the abilities to say walking with 2 legs. Chimpanzee or ape or other non-human primate can walk with 2 legs, but we have gained the ability to walk with 2 legs, known as bipeds. Second one, the ability to make tools for killing animal or hunting for food and so on. The third important thing, as a physiological thing or anatomical thing, you can say, is increase of our brain size. In the early species, you can say the brain size was as well as chimpanzee. It has gradually increased. In Australopithecus, the brain size was 35% of the modern human. In Homo habilis, it is 50%. Homo ergaster, 60%. Gradually it has increased. And it's very interesting to see our closest species, Neanderthal, the brain size was slightly bigger than the modern human. But we don't know whether they were intelligent or not. But this species is no more. But why it is no more? There are several hypotheses. One of the hypotheses given by Paul Mills from Cambridge, he said that the inherent violent behavior of modern human probably responsible for vanishing of the species. I hope most of you agree that how violent we are, right? Probably that will be true. Okay. And even if you don't believe that the genetic mutations are responsible for evolution, there are few examples. Now you look at these 3 brothers who want to be a normal parents, who typically look like the chimpanzee-like features. Their parents were not 3 brothers. Of course, I don't from Chennai, but they were in a circus company. The clandestine asked me to go and collect the sample. I went there along with very senior police official, but the circus owner did not allow us to collect the sample. Otherwise, actually we would have decoded what is the mutations which are responsible for some of the evolutionary aspects. Now you see that the mutation which helped us the way we look like today, probably there's a back mutation in this family. Therefore, the three brothers typically appear like the chimpanzee. The second one, you can see this line, so one of the primary population from Odisha. And his daughter, you can see one of the features which we have lost of body fluff here, probably again the back mutation, which again got back to the some of the features like chimpanzee and so on with body fluff. And the last one, which probably somebody is interested, you can go and look at this video. It's a Turkish family. There are 5 children in a family, 4 girls and 1 son. Only the son can walk with 2 legs, but not again steadily. He walks like a drunken person. There's no state. But the 4, unfortunately, 4 daughters, 4 sisters, they walk with 4. Again, the ability which we gain to walk with 2 legs. Again, there's mutation which is responsible for that. There's a back mutation, and therefore we're back like the chimpanzee or any non-human primate walking. I'll give you an example that if you take one chromosome, during the course of evolution, there are several mutations, there are several rearrangements of the genes from one arm to the other arm. And if you look at only the non-human and non-human primates, the human, look at some of the genes like SRY, which is present on the short arm of the Y chromosome or P arm chromosome in human. But chimpanzee, the same gene is present in the long arm or below. One over here and gorilla again. You can see that. Again, few more genes to look at. There is a delicate exospermic factor region, chromosome Y. They are all in the long arm, they are the short arm of the chimpanzee. Right? Here, this is distributed in different regions. This tells that during the course of evolution, there's a lot of rearrangement that's happened. It's just taking only the Y chromosome, and the Y chromosome has very less number of genes. And if you look at the mutation, the single base mutation, the OF1 human accelerator region is very important for brain development initially. And this gene is absent in the early organism like mammalian, fishes, and so on. But it's present in chicken. If you compare chicken and chimpanzee, the gene is present, but there's 2 mutations when compared to chicken and pig. Whereas this was about 400 million years ago. But recently, if you look at 7 million years ago, between chimpanzee and human, there are so many mutations. Okay, so that probably helped in shaping the brain development. Then there are several other genes which are responsible. Again, gene is highly conserved across the species, but this mutation in the genes are responsible for the evolution. The myosin heavy chain, which actually, again, can say that present in both chimpanzee and human, there are certain mutations which created more space in the skull. Therefore, the brain size has increased. Similarly, there are several genes have been identified as responsible for brain size. Pronotum is another gene which is essential for learning, the experience of pain, our perception, behavior, and Remember, these are some of the very unique features of modern human. Right? So this gene is responsible for all of this. Again, for the language, PARTSB2 is essential for development of languages because again it's involved in relaxation of jobs and so on. Then again, I'm coming back to this particular slide because we have seen how many species have evolved and varied. So I want to spend some time on Neanderthals, because very close species to human. Because less than 1 million years ago, these two have died, right? And if you look at where these Neanderthals have been found, mostly the Europe, particularly the Neanderthal Valley, and you can see these are some of the sites the Neanderthals were found. And this is the first skull was identified. Of course, this is a kind of model found in Belgium first, and this is a crossover cranium of Neanderthal. And there are series of publications last maybe a decade or two, and this is the first one which was done by Sartakapo, successfully isolated DNA from the Neanderthal. Initially sequenced the mitochondrial DNA. And if you look at the difference, this is how, mostly for the students, this is one of the mitochondrial DNA sequence, the other mitochondrial DNA sequence. However, the DNA is made up of nucleotides. And this side there's a G, other one is A, same way there's a G, and A G. This is how, when you compare the Rapid sequence versus the sequence of our interest, you see the variation. So based on this variation, one can identify precisely the age of the individual or the age of the population and the evolution. So here, just for giving some example, this is a good slide. This is actually Neanderthal sequence comparing with the other sequence, and based on that, we have concluded that there are about 500 to 600,000 years back, human and chimpanzee are divided. Then subsequently again, we worked on several such ancient materials, and successfully here we sequenced. There are 4 Neanderthal bones. You can see these are 88, 40, 8,000 year old samples, and concluded about this had been bridging gap between the human and other non-human other Homo species, that it bridges the gap and gave some kind of idea about human evolution. There's another species, again, I mentioned something about has extensively worked on the ancient samples. Using the same tool mechanism which we developed, that is by a small bone from Siberian cave, and he and his colleague have put the evolutionary tree. The lowest one is Davisovan, and then you can see, then the Neanderthal, and these are all human. The divergence between Davisovan and Neanderthal 1.4 million years. Right here, maybe this is about 500,000 years back. So based on this, previous studies, one can ask question whether human living in many parts of the world still carry Neanderthals and Denisovan DNA or not. This study, which sequenced 300 human genome from 140 diverse population across the world, where you can see there are a large number of authors involved, and David Leach was the one we had friend leading the study, where you can see some of the Bebo, myself, and Lance are one of this study, where we have shown that both Neanderthals and Denisovan have contributed some proportion of their their DNA to the modern. While Neanderthals, if you look at, Africans doesn't have that, whereas people living out of Africa carry up to 3% of Neanderthals. On the other hand, if you look at the Denisovan, and mostly is here, carry up to 0.5% of the Neanderthals. So why Africa did not have West? I'll come to the data that how Africa is important for human evolution. And later, group again found that this cave, the Denisovan cave, is the only place where Neanderthal, Denisovan, and modern human have lived together. And what is the proof? This study again, published in Nature in 2018, has brought a lot of evidence for Denisovan, Neanderthal, and human kind of are mixed. This study, the blue ones are Neanderthal, red ones are Denisovan, and you can see some of the modern humans, the yellow ones, carrying the Neanderthal. And in this figure, you can see there is an off Neanderthal and off Denisovan. Perfect combination to show that the marriage included between Neanderthal and Denisovan. And then again brought evidence that one point of time, all three species, Neanderthal, Denisovan, and human, lived together, and there was a mixture of genome between them. So I also wanted to highlight, because it's extensive study has been awarded the Nobel Prize couple of years back. Okay. So now I also wanted to highlight some of this study where we have shown during the COVID that about 50 megabits region, which we inherited with Neanderthal, are making individuals more susceptible against COVID. And this is on the chromosome 3. You can see that there's a signal coming from there. And this particular genome, the 50 MD region, is predominantly, you can see the frequency in South Asia. And he has highlighted, maybe this is what is giving more susceptibility for COVID infection in southern South Asia. So in fact, later on, Jnaneshwar and we have shown that no such thing as exists, because they have analyzed basically the Bangladesh people who are living in Europe, particularly in UK, because the data specifically from them, but because it has a lot more other parameters, including the height and so on. So therefore, it's not really showing that the South Asians are more prone because of the acquiring Neanderthal genome. So now I'm coming back again to highlight evolution of our own species, Homo sapiens, right? And it's very well known fact that the modern human arose in Africa about 160,000 years back. But the question is, from Africa, how did they migrate to other parts of the world? There are two hypotheses. Before coming to that, why people have migrated out of Africa. So this particular study, because it's not from biologists, this has come from geologists. So they have looked at the water level, and there was a mega between 135 and 75,000 years ago in Africa. There was no water. Therefore, people started migrating out of Africa and search for water and food and so on. I mentioned about two hypotheses. One, actually, group of historian archaeologists biologists, anthropologists, including geneticists, they believe the first out of Africa human took northern land. If there's a land connection, therefore we started moving out of Africa. But others did not believe on it because mostly the Europe, even today, climate is very bad, very cold. And those days, thinking about several tens of thousands of years back, and how they were tolerated this, then proposed the alternate hypothesis that have shown that probably the first out of Africa human might not have taken the northern route. Probably they have taken the southern coast route. All because of the existing evidence, archaeological evidence, suggests that the modern human must have arrived in Australia about 50,000 years back. In Middle East, about 7,000 years back. China, 39,000 years back. And Europe, 40,000 years back. But unfortunately, we don't have any archaeological site to give us a clue that when did the modern human might have arrived in India somewhere. There are few archaeological sites. One is a place called Jawaharlal in Andhra Pradesh. It's a group of archaeologists have excavated this site and identified a lot of stone tools, which probably the modern human might have used. Unfortunately, there was no biological sample there to analyze and conclude that when did the modern human arrive. Another couple of places are one is Kiradi, other one is ArchaeoZoology. In Kiradi, that recently a couple of skeleton was found from the children, and so it's not very old, maybe around 3,000 years old. In ArchaeoZoology, we collected recently some sample which has been kept in the museum of archaeology somewhere in India. Again, it's not to eat a good amount of DNA. The only successful story using ancient DNA for evolution study is the Arkan site travel there. I'm sure that Dr. Vashan Sinde must have spoken to you about that. So I'm not going to detail, but I'll tell you very briefly what this particular skeleton tells about. When we analyzed, we found the rocketry sample consists of two genomes. One, the other one is which I talk with later. Majority of them are from Iranian hunter-gatherers. Iranian hunter-gatherers, but not Iranian farmers. It means it's a 10,000 year old, but this sample is actually around 3,000 BC. It's not very old. Again, it's not going to give us enough information. Right? But what is very interesting here is that we don't have any Iranian farmers. There's a place called Fertile Crescent. That's where the agriculture started. Right? So there was a kind of hypothesis that the agriculture spread from the Fertile Crescent to other places. If India received or got the agriculture by Iranian farmers, then we should see some of the Iranian farmers. Unfortunately, we didn't see that, but gives evidence that the agriculture must have originated in India. That is the one point which I can mention. This is another ancient DNA study. It's actually combining ancient DNA from South and Central Asia and looking at the many contemporary samples also. Again, this was led by David Leach and we were founder. This study also is not giving much of information about our origin. So what it is giving is that there is a place called population called Yavanaya Steppe, the Pastorites, and so they have migrated from Central Asia to northern part of India, and also they have gone to Europe. And they are the one who have taken the Indo-European language, and the migration is not too old. This is only around 1000 to 2000 years. And again, it's not giving any clue about human origin in Indian subcontinent. So, in the absence of the archaeological evidence, so what is the best option available for us is to look at the contemporary population's genetics and see whether we can conclude anything based on the present day population's genetic variations. So in this process, we have looked at several populations, nearly 300, 350 different populations, and analyzed some 25 to 30,000 individuals. So in this process, we understood that the very first out of Africa human is in Indian subcontinent. Anybody can guess who this population could be? Someone says Andaman. Sorry? Australia. Not Australia. Somebody said Andaman. Yes. So that in Andaman and Nicobar Islands, there are 4 populations who typically look like Africans. Onge, Great Andamanis, Javas, and Sentinels. Sentinels are still in isolation, nobody can go near them to shoot with the arrow. Very recently also it happened, somebody tried to go there and they killed with arrow. Yes. So people try to get some acquaintance with them, but they are not really ready to come out of their own territory. So we had an opportunity to analyze them and to know that where these people are. These are the 4 groups which I mentioned: Onge, Centralist, Great Andamanis, and Java. They are very small in number. In addition, there are 2 other tribes who typically look like Southeast Asians, Nicobaris and Shanghai, like Chinese or any other Burmese and so on. And why their number is very small? They are less than hundreds, as you see here, 98, 200, and 250. Great Andaman is not 20, even lesser than that. Only Nicobaris are large number, 20,000. The reason why their number is very, very less is that when the Britishers captured India and they wanted to construct the cellular jail, I know about, if you know the history, our freedom fighters have been taken to Andaman and Nicobar Islands and they have been put in this jail. It's called as cellular jail. And the way there are 7 wings, as you see, I think 1, 2, there are 7. I think 2 are still down, 5 are still there. And each one has a window, backside window of one of the cells. So many cells. That is the front side. When somebody, freedom fighter, is inside the cell, they cannot actually see anything outside. From here, front, they can see only the wall. Backside, they cannot see anything. So that's the way our freedom fighters have been kept. The reason why I am saying is that while the Britishers want to construct the cellular jail, most of the tribal groups, they were coming to protection, like the way they have started killing. That time, the Britishers used their own force and killed lot of them. And after killing them, it's very interesting that they have taken the skull and hair samples and they kept in the Cambridge Museum, the Network Laboratory Museum. In fact, that allowed us to collect some of the samples. Along with that, of course, we could not do much of this, but we had an opportunity to go and collect the sample from Andaman and Nicobar Islands, particularly from 3 tribal groups. These are Onge, Great Andamanis, and Javas. Central is still there in isolation. We could not go and collect the samples. And we use different markers not only for this study and subsequent studies. In the genome, as all of you are aware, there are two different kind of the cell, two different kind of genome. One is the nuclear genome, which is equally contributed by both the parents. We have 46 chromosomes, 23 we get from the mother in the form of egg, 23 from father in the form of sperm. The non-organization again becomes 46. While we inherit the nuclear genome equally from both the parents, we inherit the mitochondrial genome only from the mother. Okay. Every cell, while we have only two copies of nuclear genome, there are several mitochondria. Each mitochondria has several mitochondria. That means two copies versus hundreds of copies of mitochondria, which exclusively come from the mother. So, we wanted to trace the maternal lineage, we use the mitochondrial DNA. And if we want to trace the paternal lineage, we use the Y chromosome. Because Y chromosome also goes to male lineage from grandfather to father, father to son, son to grandson. It goes without much changes. Therefore, this is a good marker for tracing the paternal lineage, and this is a marker for tracing the maternal. So, we used the complete mitochondrial DNA sequence to understand the evolutionary history of the animal tribes. This is called as haplogroup tree, worldwide haplogroup tree. And you can see that every country or continent are with either exclusively one particular haplogroup or few haplogroups, or predominantly those haplogroups are present. For example, take Africa, you can see all the L-direct haplogroup, L1, L2, L3 are found here, but you don't see that in other places. Similarly, Europe has predominantly one haplogroup. And you can also see India, they have haplogroups. Similarly, you can see Australia, India, and North and South America are predominantly one particular haplogroup. So these are defined by a set of mutations. Mitochondrial DNA is 16,500 base, 16,500 bases. And there are certain mutations which are evolutionarily conserved, doesn't change frequently. So we use those to construct these haplogroups. When we try to look at the mitochondrial DNA of the animals, you can see that they have unique set of mutations. These numbers are the position of the mutation. They did not fall into any of the known haplogroups which has been identified so far. So based on our observation, you can see that these many number of mutations are present only in Andaman but not in others. As I mentioned earlier, but we can also calculate the age of this population based on this mutation. It turned out to be 65,000 years. So that's most of the work also done by Vigyan, as you see this one here. And based on this, we propose that the Andamanis are the first modern human who were migrated out of Africa, taking southern coastal route and reaching Andaman and Nicobar Islands about 65,000 years. So this is the first genetic study which shows that the Andamanis are the first modern human who were migrated out of Africa and reached Andaman and Nicobar Islands. Of course, the study was published in 2005. 20 years has gone, but no contradiction so far because this is still so true that this is the first modern human who are migrated out of Africa. And they didn't stay only here. In this process, they stayed in southern part of India, some stayed here, some further migrated to Australia. In fact, the Australian Aborigines, some of the Indian tribal groups were Kerala, Kurumba, and the Andamanis still have some kind of genetic abilities. So this shows that Indian subcontinent has the very first out of Africa human. Then we used the nuclear genome after human genome was sequenced the variations present across the genome and which are highly polymorphic across different population groups have been identified. This is 1 million SNP is available as a chip which we use to look at the population across India. And where we put the representative population from all the linguistic groups. We have 4 major languages, as I hope you are aware. Dravidians, Indo-European, Austroasiatic, and Indo. We also used population from different social groups, so-called upper caste, middle caste, lower caste, tribes, and primitive tribes, to have representation from all the linguistic group or social group, and also representing all the geographical groups. So in this study, in fact, we constructed the Indian population history. So what we have shown was that in the pre-Islamic India, there were 2 founding populations. One we call them as ancestral South Indians, other one is ancestral North. The ancestral South Indians were the early humans who were migrated out of Africa, while the Andamanis were going to Andaman and Nicobar Island, some stayed in southern part. Ancestral North India were migrant, again migrated out of Africa, they migrated towards north. There was split somewhere in the Middle East. One group has gone to Europe, other group has come to northern part of India, we call them as ancestral North. There are several examples to show this abilities. One is the skin color mutation. Chandana's for on that extensive. Other one is the lactate persistent, which are common in Europe, Middle East, and northern part. And white chromosome haplotype R1A1A is again common in Europe, Middle East, and northern part of Africa. All are suggesting that they have chromosome. These are the 2 founding populations. On the other hand, if you look at these principally from PCA plot, these are all Indians. The European have not samples, African and Chinese have not samples. There are several individuals, but still they are in a single cluster, suggesting that genetically they are more homogeneous. Because of that, they are in a single cluster. Whereas look at Indians, every single population is unique. Although every single population is unique, but still they all share some proportion of genome. Therefore, they are not clustering somewhere here and there, but they are all in a gradient of cluster. Although every population is unique, but still there is kind of genetic mixture within the population. So here I mentioned about 2 founding populations. These 2 founding populations have admixed and gave rise to many population groups. For that I come bit later, but what I want to highlight is based on our finding, we also recruited the Aryan invasion. And based on some of the findings which came very recently, there was a big writer by Tony Joseph. He mentioned that the problem of Aryan migration has solved because R1A is common in Europe, Middle East, and another part of India. In fact, Jnaneshwaran and myself, we wrote a refuted for that. That is not too easy to resolve about Aryan migration, and this is not more things we study. So this shows that in fact Aryan invasion theory, although we refuted early, and this is again very confirmed. So that's one aspect, that the founder population in India are the ancestral South Indian and ancestral North. But in addition to that, there are several migratory events happened in the recent past. With the Alexander army, come to Turks, British colonization, and very recently cities, Jewish, and also Parsi population. To give you some idea about that, to take the cities who migrated recently about 20 years back, they are also in Hyderabad and also in Thangaraj. When we look at the genome, three different genomes. One is autosome, mitochondrial, and Y chromosome. These are Africans, Indians, and the Sindhis. These three different genomes have three different proportions. You can see Y chromosome, more of African, whereas Indian, and mitochondria, less Y chromosome, more mitochondria. Because actually goes with the historical aspects of the migration of this Bantu-speaking Africans who have been brought by Portuguese traders, and they have been, mostly the males have come and they have been sold to work in the army of now and Sudan. So that shows that so very few numbers have come and they vary globally. Other one is the Jewish. Again, they have analyzed mostly they are preserving their Jewish genome or they are mixed mostly. And their original genome is only this much as you see here. And mostly they are mixed with the local Indians. And in this process, we also looked at some of the mutation which causes the cancer. And in this process, they have actually caused the original mutation which is responsible for the causing cancer. And this is about Parsis again, Ganeshwar's work, where unlike Jewish, they preserve their old genome, and that's why, because of endogamy, they have not more diseases happening in Parsis. Right? This is just some of the highlights which I want to talk about. Very recent migration to give you kind of idea about the Indian population. So all of them put together, one can say, it's not exaggeration actually, to say that India is an ethnic museum because we have a collection of large number of population groups. We have 4,635 population, large number of tribes, and tribes are very few. They primitive in the sense they are very primitive. You cannot actually now call this as a primitive tribe. Most of them tribal. And 36 of them still practice under a gather. They just go and hunt for food. They don't do any job, including agriculture. And if you look at broadly how the Indian populations are, there are several, it's very difficult to read. The first wave of migration, then there are several subsequently migrated, and if you also look at the social structure, then probably they all evolved from the ancestral population. So this broadly gives about the Indian population structure today. Now I'm coming back this slide to highlight that I spoke about ancestral South Indian and the ancestral North Indian, and there were adventure between these two groups. After that, there was several populations which have evolved. To understand when this adventure took place, took another study, and you can see there's large number of populations south, large number of populations north. And based on the recombination between the homologous chromosome, can estimate this, and we found that both ancestral South Indian and ancestral North Indian initially gave rise to many populations. And if we just go back and imagine that the initial population migrated and settled in the jungle where they had enough of food. After some time, there's no food is available. Those who are active must have migrated from one jungle to another place just for food, and from there to another place again. So that's the way initial migration must have took place. And once they settle in a place, they start marrying only people available there, and those who are in neighboring forests, maybe it's not too far, maybe 10, 20 and 50 kilometers away. There's no connection between these two, therefore they are in isolation. They acquired mutation. That mutation distinguished from other populations. Okay, so that's the way it happened. But this migration probably took place until last 4,000 years. In the last 2,000 to 4,000 years, there is an admixture between these two founding groups. Okay? And the color indicates of the admixture, the color between these two groups. And this happened for 2,000 years. After that, last 2,000 years, every single population started maintaining the environment. Means marrying. So this is what the present day scenario. And this one is very clear that I take the best example for this admixture is skin color. Nowhere in the world you see such a range of skin color. We have extreme light skin color, we have extreme dark skin color, in between we have different shades. Right? That itself shows that we are admixed. And this admixture, as I said, stopped, and the last 2,000 years, every single population started making the indoor land. Indoor means married within the group. From the caste system, which arose much later, maybe around 1,000 years back, was to put more pressure on every single population to maintain the indoor land. Now let's slightly go away, but it's very important in terms of public hearing, is that how the endogamy has influenced on our health disease. So to study again, now we extended the study and included population from other South Asian. The reason is that whatever we do in India is also relevant for other South Asian countries, because India contributed genetically at least 50% to other countries. And some of the mutations, particularly, I put the slide but I deleted it because I don't have time. There's a gene called myosin binding protein C3. We found 25 base per deletion in the gene, which causes a disease called cardiomyopathy. So that mutation, when we looked at worldwide population, we found predominantly in India and also other South Asian countries, but not in other countries. Therefore, it's important for us to look at other South Asian countries also. We collected 2,800 individuals from 275 population. You can see again representing all the major linguistic groups, social groups, and so on. Again, we used 1 million SNPs. So in this study, interestingly, what we found was there are large number of population in India which are expected to have population-specific mutation, which might cause population-specific recessive disease because of the practice of indoor land. Okay. When there's a mutation which is dominant, that means in a pair of chromosome, one of the chromosomes carries the mutation. If it is a dominant, obviously those who are carrying the mutation will have the disease. But if it is a recessive mutation in a heterozygous condition, that means one of the pair has a mutation, doesn't cause any disease. It's a recessive. Our main worry is that because dominant mutation causes the disease, recessive mutation doesn't cause any disease, but the potentially in a social structure like where individuals marry within a community, the mutation might have arose sometime back in an individual. He or she who carry the recessive mutation will be absolutely fine. But when he or she falls on the next generation, the again next generation, goes generation to generation, then it is widely distributed, and there's an opportunity that two heterozygous individuals who are absolutely fine to marry and give rise to the homozygous mutation to the next generation, therefore there's a disease. So that is very, very common in India. The kind of analysis, I don't want to go into detail to give you the statistical information, but look at this IVD scope. There are several lines. Every single line represents one population. There is also a couple of lines here, which is in the color. One is as Jewish, other is Finnish population. Right? You all know these two populations have been mapped for a large number of diseases because they are following the heterogeneity. These are all diseases, these are the genes, and most of them are autosomal recessive. Only few of them are autosomal dominant in a Jewish community. This is Finnish population in larger provinces. When you have situation like this with very tiny IVD, so there are large number of disease, and you can imagine what is the consequences of large number of population in India, just several fold higher IVDs. In a simple way, what it means. The IVD is calculated, for example, you are taking 100 individuals from a company. If you say that all the 100 individuals are unrelated, then you take 10 kb regions, compare them, you would expect that in the 10 kb there may be lot more variations, right? But out of 100, 50 have come from a common ancestor. Then today they are unrelated, but still all the 50 will have the same haplotype. There's no difference. That itself states that today they are unrelated, but actually they have come from a common ancestor. Okay, so that's what is indicates. So how does it actually matters for anti-leukemia? Look at this pedigree. It shows that I hope that all of you are aware of the pedigree. Female, male, they are married, they have two daughters, right? And here there is a mutation in a heterozygous condition. This is a recessive mutation, therefore there is no disease here. She married somebody who doesn't have the mutation, and therefore it goes again the heterozygous condition to both of her daughters. And both the daughters marry, not going to outside of the mutation. Then again, going generation after the generation in a heterozygous condition. Therefore, no problem at all. Come to this generation. Now, or even you can go to some more generation. Both the girl and boy who carry the mutation heterozygous, they are absolutely fine. But in the same community, they want to marry. There is statistically 25% of chance that both contributed this abnormal chromosome to the next generation. Therefore, there is a disease. Okay. So this is what is the prediction. In fact, we found earlier in one of our studies where you can see it's a consanguineous family, of course. Both the parents, the boy, the male was 45 years, father, and the mother was 10 years, absolutely fine. And you can see the electrocardiogram. This is a white type C. He is a mutant. The parents are with heterozygous, both C and T overlapping. The one chromosome carrying white type, other chromosomes with other mutations. And in this case, unfortunately, this is a gene called polymerase I. This is a nucleotide essential for mitochondrial DNA replication. Because of single base mutation, it introduces multiple mitochondrial DNA deletion. This is a white type. You can see multiple mitochondrial deletion. And because of that, it causes encephalopathy, seizures, and stroke-like deaths. And most importantly, children who carry the mutation, they die at very, very early age. You can see showing that 13-year boy died, and this child from must have died when I analyzed the who are actually carrying a homozygous mutation like this gene. Fortunately, only one child out of the three is with the heterozygous mutation, and we have to look for this future of this particular child that he should not marry somebody with the same family with this mutation, or even generally we have to do the diagnosis to avoid the child carrying homozygous or previous carrying that homozygous mutation. This is one example. Again, going back to this slide, we don't have any idea when we study this many number of population. We collect this sample for looking at the population study. Because now we identified large number of populations, or at least 1/3 of the populations are expected to have population specific recessive mutation. Therefore, we selected a few populations from here to see really they have any problem or not. Right? We selected 4 different populations, and when we looked at them, at least 3 we found a problem. One population is reddies. They have high frequency of HLA mutation, which causes ankylosing spondylitis. They are from one particular. Another population, they have mutation, high frequency mutation in gene, which lead to cardiac issues, which we are now. The third population, to our surprise, we found, when we went back to the population, we found high frequency of this. This is called junctional tendency epidermolysis bullosa. This is skin rashes. Also, children have the congenital heart disease, and they don't survive beyond 2 months. They die within 2 months after the birth. But unfortunately, when we go to the population, you don't see anybody surviving with the disease. But the parents are carrying. Obviously, it looks like the population is healthy, but the parents are carrying this mutation. When we did the exome sequencing, we found the 11 base deletion, the G called LAMP. And as the pedigree shows, parents are heterozygous, children died. Then we looked at in the 10,000 genome sequence, which we completed, to see whether how it is distributed. And just show that this was the releasing of the 10,000 genome, and the Honorable Prime Minister actually announced that India has completed 10,000 genome, and none of them had the mutation in 10,000. Then we looked at the worldwide database consisting of altogether 8,62,000 individuals. If you look at the chromosome wise example, 70 lakh chromosome doesn't carry except 2 individuals with heterozygous mutation. That means it's absolutely rare, ultra rare mutation, which is present in a particular population. And if you recollect that endogamy actually plays a role in having this high number. Because it's absolutely novel mutation, we don't have any clue whether it's really causing the disease or this kind of association. To do that, we have to create animal model, generate an animal model, using CRISPR-Cas based, and this is the strain which we use, deleting precisely this level CRISPR. And unfortunately, the pups are not surviving beyond 2 days. They die within 2 days, but within the 2 days, we can see there's a mark where scaling of the skin. Probably here you can see better. You can see there's indication that there's a kind of scaling is happening. But unfortunately, we could not do anything beyond this. Whereas then we looked at, because one of the issue is the cardiac problem. We took the heart, tried to look at how the heart structure is. Using micro-CT, this is a white type, the first one, this is heterozygous, both are similar. But in the homozygous condition, one is very big. But interestingly, we found there's a certain defect, there's a hole within the subcontinent. That could be one of the reasons that why there's a death of children and also this animal within 2 days. And the cross section again, you can see these two are alright, and the homozygous is showing the hypertrophy. The chamber became very, very narrow. So we could not do anything beyond this. Then fortunately, in this process, because sometimes the crystal gas is off target, and we got a clone with 45 base pairs that include this 7 base pair also. When we looked at how it is impacting the amino acid, while the precise 11 base pair deletion terminates much before, so it removes 373 amino acid, whereas this, although the deletion is much longer, it removes only 15 amino acid. Therefore, the impact is not much. So again, we allowed the animal to grow. Up to 4 months, there was no problem. But after 4 months, slowly the symptoms started happening, and this is after 4-5 months. And in 6 months, the animal dies. Look at the internal organ. There's also edema, dissected out all the organs. You can see the hypertrophy, spleen is enlarged, stomach becomes very small, but the animal doesn't eat. And internally, we put the microscopy and found there's a block in the stomach. So all of this happening because of this 11 base deletion, which leading to all kind of abnormality. Unfortunately, the children die at very, very early age. Now, for our understanding, scientifically, this is fine. But whether we can use this to help the family, then went back and found that there was a mother who already lost, brother is pregnant, lost 2 children because of the mutation. Then we brought the mother, actually mother was already living in Madurai, here Madurai. We brought the Madurai to Chennai, collected the organic fluid and tested whether the fetus is carrying heterozygous mutation or homozygous or wild type. And found, I don't know here, but here you can see this is a homozygous, heterozygous. There are 2 alleles here, showing that it's heterozygous. Again, we found by sequencing, again you can see it's heterozygous. Mothers and the grandparents. And we advise the mother to continue the pregnancy. The child born about 100, maybe 1.5 years back, with a proper time, and it's growing all right. At least it gives some kind of satisfaction that what we have started about 30 years back, looking at population history, now it is helping the society. Okay, this is one family. So we don't want to stop with this. We want to screen the entire population. Right now, much bigger challenge. We are approaching the government. We approached there's a Directorate of Public Health and Preventive Medicine in Chennai. We approached them, and after year-long paperwork, they gave us the permission or approval to go and collect the sample from all the 5,000 individuals. And this is the last camp which we had, the month of July 19 and 28 July. Along with the partner, we collected the sample and tested and found about 5% of them are carriers. This gives you idea that around 5% of the individuals who carry the mutation, they don't have any clue, but suddenly they say that they marry another heterozygous individual, suddenly the child is born with this kind of, So it's going to actually help once we are successful with this, then we identify several such populations. Having a population-specific mutation, population-specific disease is going to be helpful for all those populations. So now, just wanted to highlight how important we begin with how important the evolution is. Again, we wanted to highlight. Okay, this is not our study. Somebody has shown that how important to study the evolution. This is a summary of the study is that our general conclusion is that evolutionary biology is a crucial basic science for medicine. And below, so that evolutionary biology is not just another topic vying in inclusion in the curriculum. It is an essential foundation for a biological understanding of our health and disease. Okay. So now I'll shift gear to ancient DNA study, because it's also your curriculum, right? So ancient DNA is a DNA which is isolated from the organism which is known to have lived in the past, but there's no time limit for that. It could be of several years or several hundred years or several thousands of years. And using ancient DNA, what we can understand. So what type of plant or animals lived in the past, what type of food extinct human or animal ate. There's a way of looking at using carbon dating or using many other technology. So how similar, particularly genetically, the extinct plant, animal or the human species today. What animals were hunted by the ancient humans, because when they eat, there's a tissue which is there, can be isolated and understood. What type of disease was there in the ancient people, and establish the genetic affinities of the living people. These are some of the utility of that, and beyond this there are many other utilities which I come to. So what is the challenging task for the ancient years? It's not like taking the fresh sample and utilize it. It has its own challenges. There are several breakage of DNA, oxidative lesion, DNA cross-linking, hydrolytic lesion. All of these gives fragmentation of DNA as you see it. And ultimately, one is supposed to see as a high molecular weight here, DNA is fragmented because of all these conditions. In addition to that, there is also contamination with fungi, bacteria, and so on. When you isolate DNA, you see the DNA. Of course, you don't see the interact DNA, but you can also see sort of the micro DNA from microorganisms. So therefore, it's very important to have the ancient DNA lab, and this is the process that you have bone or tooth, eat that and make the powder, and ancient DNA samples are not like the contemporary samples. You can just use the standard protocol. So every sample is unique because of the age of the sample, because of the nature of the sample, plus modified declassification, many more. And of course, once you get the DNA, then we use for sequencing and so on. This is another challenge where you have endogenous DNA, this is exogenous, and this is a contamination. And the way there's a way of amplifying it by using the adapters. Of course, once you sequence the contaminated DNA can be eliminated and capture only the human sequence. So because of the complex nature of this, the ancient DNA laboratory is essential to handle this. You cannot use the general laboratory because otherwise you will contaminate your own DNA. All those who are handling will contaminate. So laboratory under positive pressure in the room or the whole lab itself have positive pressure, and usually is on all the time because they come on the DNA which is existing in the sample, and there's a one-way traffic because when you go from one direction, go out from the other direction. These are some of the basic things about the DNA, ancient DNA. But I will give you some of the examples how the ancient samples are helpful for understanding some of the very basic things. This is one of the study which wherein initiated by David Reach and many other groups, we are part of it. They have analyzed 3 different bone samples. 8,000-year-old from hunter-gatherers from Mandala, 7,000-year-old from Zagred, and again 8,000-year-old from Lesotho. These are some other ones. So this had actually helped in understanding the origin of Europeans. As we have shown, in India there are 2 founding populations. In the Europe, they found that there are 3 founding populations. And these are the bones actually being collected. And these are 3 different directions. Of course, as I mentioned, David and Saurabh again there, myself, on the single part of it. And what is the conclusion about this study, which I wanted to highlight? That they say that there are 3 populations. One is indigenous hunter-gatherer. Second one is Middle Eastern farmers migrated to Europe around 7,500 years. Third one is North Eurasian and genetically connect Europeans and Native Americans. Okay. So what is interesting point here to observe is based on the mutation, what their skin color would be, what was their perspective in terms of amylase gene and so on. Early farmers had lighter skin color. Their skin color arose when population moved out of Africa, and their bones carried that mutation. Therefore, one could predict that the early farmers had a lighter skin color. The second one, both the hunter-gatherers and the early farmers had high copies of amylase gene. The amylase gene copy number increased after agriculture came into existence. Right? That means they had a high reach of farming product, therefore they acquired more copies of amylase. So none of the ancient human was able to adapt to digest the milk sugar. Right? So this mutation, lactate persistent gene mutation, arose around 5,000 years back. But you can clearly see these are 7,000 and 8,000 years old bone samples. But the animal domestication happened much later. After drinking of domesticated animals, we acquired the mutation to digest the milk sugar very efficiently. But they are not trained because that was only 5,000 years old. But bone samples are 8,000. You can now imagine how the ancient DNA sample can be utilized. Now I will tell our own couple of studies. One is the Rupkund study, and this has some of the videos you can look at it. Rather than me explaining that, I think the story from the video will be much more easy. Later. Mouse can be here. Was active. We hope the audio is good here.
Looking at Rupkund, it is not difficult to understand the dangers involved in either climbing or passing through. Rupkund Lake looks beautiful, in fact stunning. There is clear, sparkling water. The hills around the lake will provide shelter, but the appearance is deceptive. Rupkund doesn't look pretty anymore. It resembles a graveyard. Skulls placed on the stones remind one of the tragedy that occurred here centuries ago, of the misery, of sadness, of the helplessness of the weary victims who died a slow, painful death. There are hundreds of bones strewn all over the place. At some places, the skeletons are stacked as if they were washed away and blocked by some boulder. Pile after pile, the bones and skeletal remains of human bodies, flesh darkening and hiding away in the ice.
Yeah, so to give you idea, this is a lake. But anyway, to give you some idea, this is the lake which is in the Himalaya, about 5,000 feet or I think, meter above the sea level. And this is a place. But anyway, we'll move on to that. And if somebody has to go, they cannot go straight because their body has to accommodate. And anyway, we'll move to the next one. This gives you.
Since the mid-50s, several people have explored the area and offered theories on the remains. So who were they?
This tells that who are these people.
Yes, great.
What were they doing there so far from habitation? And where were they going? No one has yet solved the riddles of the dead. Were raccoons dead a lost army felled by the blizzards and landslide? Were the pilgrims seeking to get closer to the goddesses not awaited? Did a large group of people commit ritual suicide here?
These are some of the
Could it be that
speculations.
traders
Who are
got
these people?
hopelessly lost and perished? Were these the victims of a terrible epidemic? No one knows for sure.
So these are the speculations. The ancient, there could be ancient traders on their way to Tibet. Second one is Zorawar Singh's army from Jammu tried to invade Tibet in the year 1841. King Jaswant of Kannauj and his wife and attendants went there and something happened, so they have perished. This also some kind of speculation that there was a victim of the epidemic, and that's very, very strange that why they would carry all of them up and dump there. Then victims of ritualistic suicide, this is also a very strange thing. But actually, to understand who are these people, our colleagues from CCMB, they went there to collect the samples.
The team of Indian scientists leave Varuka after months of planning. The team leaves to Walla from Delhi early morning. The drive to Walla was told to be extremely tricky. The team finally arrives at Woodcote. There are no signs of any tragedy. From the top, there is an excitement as the team unearths the body of a woman. Not too deep in the snow, the body lies face in.
Yeah, so this just to give you some idea that there are hundreds of whole samples, and we collected our colleagues. When we looked at, there are two distinct set of samples. One is about 800 age is 800 years, other is 1,800 years. But we used various markers, mitochondrial, Y chromosome, and other nuclear markers. We found there are three clusters. So one group is basically Indians, what you see here. There are Indian samples, the gray color one, and this is superimposed, the red ones. Okay, that's one. Second one actually looks like the Greeks and so on. That's why they're all Mediterranean. The third one is only one sample looked like they are Southeast Asian. So that means out of this some 20, 30 samples, there are three groups. We did not find any of these speculations. Right? Like these traders could be because the Mediterranean were migrated, but why they have migrated? And there was no king artifact also not there. And only one sample showed they are Southeast Asian. There are hundreds of samples. This is only 20, 30 samples. I think if we analyze all of them, probably we may have better conclusions. So this is one study which I want to have. So other one, we talk about there's a Queen Ketevan, and again this video will give you about the entire story of this Queen Ketevan. Please watch it.
Queen Ketevan, great-granddaughter of King Constantine of Carthage, ruled over Georgia in Eastern Europe. In 1630, the Emperor of Persia, Shah Abbas I of the Safavid dynasty, conquered her lands and threatened to destroy Georgia. But the queen, to protect her subjects from annihilation, surrendered and was taken prisoner in 1614. She remained a prisoner of the Shah until 1624, during which attempts were made to convert the queen to Islam and force her to join his harem. Both bribes as well as torture were used to persuade her to relinquish the Christian faith. Queen Ketevan, however, withstood all the attempts and was finally killed on September 22, 1624. Her death is also mysterious, as one account says she was crucified to a tree, while another says she was strangled. Legend has it that a light appeared on her body, and the patriarch of Georgia declared her a martyr and saint. The story, however, doesn't end there, and it is believed that two Augustinian friars, her confessors, discovered her remains in Shiraz and brought them to Goa in 1627. It is these relics, her right arm and hand, that have come under attention, as it is believed that they were buried in the chapel chapel of the Augustinian convent and may still be there.
Research of St. Ketevan's relics started in 1989. However, efforts in earnest began in 1998 when the Catholic Patriarch of Georgia wrote to the Ambassador of India in Ukraine, seeking permission to search for the relics of the Queen and Rome. Subsequently, a Georgian delegation, which included a bishop came to Goa, but failed to locate the remains.
Silva Grego relied on the manuscripts the Augustinian tribes written in 1870, 1723, and 1629, and in them, in a scientific manner, described in detail the tombstones in the church floor, the side chapels and the chapter chapel.
Using archaeologically sound techniques, the ASI has cleared the ruins, and in their labels have recently chanced upon the most likely resting place of these relics.
This is the second window on the epistle side of the order of the Catholic Chapel, about which, according to the Book of Cellular, who once entombed the remains of Queen Ketevan.
Entombed in the black box, along with Queen Ketevan's remains, were the relics of Fray Guillermo Padmashree and Father Geronimo de Cruz. The ASI came across remains of the black box and a longish bone fallen below the second window. Can this bone be the right arm of the saint?
So this was a question asked by the Archaeological Survey of India. So we started this work long ago, and we can really see whether this bone belongs to Queen Ketevan or not. We sequenced and looked at the 22,000 individuals of human Indian population, but we did not find a specific sequence which we found in the bone sample. Then we looked at whether it's a male sample or female sample. This gene called apelogeny. In the male, you get two alleles, female only one, so only one shows it's a female sample. Then we asked them to collect some sample from Georgia. They got some 30 samples. 2 out of 30 are matching with the bone. Okay, so these are the 3 evidence suggesting that this bone probably belongs to Queen Ketevan. This was published on 2014. But Georgian government want to take this bone because they consider the Queen is one of the sacred. Therefore, between the government it was happening, and during COVID time, 2021, our SRL Minister Jayashankar has handed over part of the bone to his counterpart in Georgia. And this has been considered one of the important events. In fact, Honorable Prime Minister came in one of his An-Ki Baat program on 21st July, was describing about that. In fact, he has appreciated his work for research and academia and so on for solving this issue. So you can imagine that not only a research point of view, it's helping in terms of between countries and many other aspects. I didn't talk about the use of ancient DNA in forensic and so on because of time limit. I have to rush now. I think this is the last one, and thank all those who have contributed to my own group and many others in other institutes, and the funding has come from different sources. And thank you all. Sorry, I don't have time for interacting with you. If necessary, we can have online discussion sometime. I'm sure can organize it. And, thank you very much.
Yes. Thank you very much, sir, for coming and enlightening our students, especially from the ArchaeoZoology and others of faculties of zoology and other departments who had come here. Thank you very much, sir, for this enlightening lecture. A round of applause from everyone for his.
online interaction or through email if you have any questions regarding today's lecture. Thank you very much. Thank you all.